Hello, my name is Carol Leibel, and today we're going to look at the development and the influence that genes have on this process, part 2. So last time we were together, we were using the fruit fly as an organism to examine the process of development. So we have an egg, a fertilized egg, and we call that a zygote. Now, inside that egg, there are indeed messenger RNAs from the mother, and these are called maternal effect genes. And so there are messenger RNAs from those genes. And now what will happen is that mitosis will take place. And in this particular part of the diagram, approximately 9 nuclear divisions have occurred. And they happen without cytokinesis. So it's kind of like a bag of nuclei, if you will. And it's called a syncytial blastoderm. Now, after 13 nuclear divisions, the nuclei migrate to the periphery of the embryo, if you will. And now what will happen is that cell membranes will form around each of those nuclei. Now, even at this point in time, the body axis of the little embryo has been established. So at this end, that is going to be the head, the anterior end. And over here, that's going to be the posterior end. Up here, this will be the backside or the dorsal, and opposite of that will be the ventral or the tummy side. And then we'll have also left and right. Once those cells have been formed, then this structure is called a cellular blastoderm. The next step in this process is that this particular blastoderm is going to be divided up into segments, kind of like an earthworm. Now, those segments are divided up based on protein gradients that we're going to talk about in a few minutes. So when that segmentation takes place, this becomes a larvae. Remember, we mentioned that there are actually 3 stages of larval development, and we call them instars. So there's a first instar, a second instar, and a third instar. The next thing that will happen is that segmented structure is going to enter a pupa, if you will, and those tissues will be broken down and regenerated, and then what will emerge is an adult. There will be 3 main parts to that adult: the head, the thorax, and the abdomen. And each one of those, with the exception of the head, is divided into segments. All right, so let's look at this again. So remember then, we have this little zygote that underwent mitosis. This is a syncytial blastoderm. There are lots of nuclei inside of there. And we can already see the establishment of gradients. We talked about before the bicoid protein gradient and the nanos protein gradient, and then interacting with that, we also have hunchback and we also have caudal. And those 4 protein gradients are going to establish the anterior and posterior ends of the blastoderm. Now, once that has occurred, the next thing that's going to happen is that we're going to divide that blastoderm into 3 major parts, if you will. And those are going to be controlled by something called gap genes. And mutations in these gap genes are going to create a malformed larvae. Okay? These gap genes are going to encode transcription factors, And these transcription factors are going to directly control the expression of another set of genes involved in further segmentation of the larvae. So these transcription factors that have been produced by the gap genes are going to activate another set of genes called the pair rule genes. And so if we look at this, we can say this is segment number 1, segment number 2, and segment number 3, and so on. So all the odd segments are controlled by a certain set of genes. And then all the even segments, which are the light blue ones, I'm going to put X's on those, are controlled by another set of genes. And so together, these collection of these genes are called the pair rule genes. And they are responsible for dividing the blastoderm into 14 different segments. Now, it also turns out that these pair rule proteins that are made by the genes also are going to act as transcription factors. And what they are going to do is that they are going to activate what we call a set of segment polarity genes. And these segment polarity genes are responsible for taking each one of those segments and establishing which side will be the anterior side and which side will be the posterior side. So these genes, these segmentation genes, are responsible for dividing the blastoderm up into various segments. This is what the larvae looks like when it's normal. There are 14 different segments. Okay, and as you can see, when there's a mutation in any of those genes, it results in a malformed larvae that doesn't have all 24 segments, if you will. And if you're looking here, these right here are abdomen segments, and this right up here are going to be thoracic segments, and that right there will be a head segment, if you will. Now, these genes were discovered by two wonderful scientists, Christine Nussel-Valhard and Eric Weishaus. And what they wanted to do is they wanted to identify each one of those segment genes that we just talked about. And the way that they had to do that is that they would take the Drosophila egg and they would basically submit it to different kinds of mutant-causing chemicals, and then they would look for deformed larvae. Once they found those deformed larvae, they would systematically characterize their phenotypic mutations and identify the gene responsible for that development of the axes. So they were able then to identify which genes had gone awry. They were able to identify 15 genes that, when mutated, caused defects in 14 segments of the fly embryos. They further classified the genes into groups based on how and in what order they affected segmentation. As I mentioned, first they identified 3 gap genes, which helped determine the identities of each segment of the embryo. Secondly, they identified 6 pair rule genes, which affected the alternating segments, the odds and the evens. And then finally, the group identified as 6 segment polarity genes that helped establish the anterior-posterior axis of each segment. All right, so they were responsible for that. Once we had each one of those segments laid out, the next thing that's going to happen is specialization of each segment. In normal flies, each segment is going to have different kinds of structures like legs and wings and antennae. And this process requires the action of a group of genes called homeotic genes. Edward Lewis in the 1940s was able to discover these homeotic genes through mutants. So the mutant flies, in which the structures characteristic of one segment of the embryo were found at some other segment, like you'll see in a moment, there were indeed legs growing out of the heads of flies where there should be antennae. So let's look at what is a homeotic gene. Just real fast, this is a picture of Dr. Lewis. He passed away in 2004. However, in 1995, he received the Nobel Prize along with Nusslein, Volhard, and Fischhaus for development in Drosophila. So at any rate, these genes were that he discovered were dedicated to the development of each of the segments of the body. And by causing mutations in certain genes, he found that he could cause the flies to grow extra body parts or other abnormal features. So these were called homeotic genes, and they regulate the development of anatomical structures in various organisms, and they are found in insects and mammals and plants. So homeotic genes encode, it turns out, encodes for transcription factors that control the expression of certain genes responsible for those anatomical structures that we just talked about, like the wings and the legs and the antenna. Within each of those genes, there's a sequence that is 180 nucleotides long, and they code for 60 amino acids within that protein. And that particular area of the gene is called the Hox gene. And that particular part of the protein is called the homeodomain. So let me show you what that might look like. Okay, so this would be a homeoprotein. And this little area right here has 180, not 180, but 60 amino acids. And it's this part that is responsible for interacting with a particular gene and activating that particular gene. So that particular area of the protein is called the homeodomain. And as I said, they act as transcription factors that activate certain genes responsible for certain characteristics. So the Hox genes are switched on in different segments, and the pattern of Hox gene activity give each segment an identity, telling it where it is in the body and what structures should grow. For instance, genes that are active in the head would perhaps direct the growth of mouth parts and antenna, while Hox genes that are active in the thorax would direct the growth of legs and wings. Drosophila have been identified with 8 Hox genes, and it turns out that all insects have 8 Hox genes, and they are clustered into 2 complexes, both of which are located on chromosome number 3 in Drosophila. Now, it does turn out that these Hox genes are arranged in clusters, and their order on the chromosome is the same order in which they appear along the body. In other words, genes on the left here control structures found on the anterior portion of the body or in the head, whereas genes over here on the right-hand side control patterning found in the tail, maybe for legs. changes to a Hox gene expression change the segment's identity. For example, in the first segment of the thorax, normally grows legs. The second Hox gene grows legs and wings, and the third grows legs and halteres. So when a Hox gene activity in the third segment is made the same as it is in the second, what will happen is that segment will grow legs and wings. That sounds kind of crazy. Okay. All right. So here are some mutations. On the right are flies that have normal development. So for here are where we would find the little antenna. And there is a mutation that occurred in the Hox gene. And so instead of that protein activating the antenna gene that as a transcription factor, what it did is it activated the gene for the development of legs. And so now right here where there should be antenna, we can see legs growing. Down below, here is a structure called a haltere. And so the gene that, the Hox gene that is responsible for the development of that particular structure, if there's a mutation and that Hox gene now activates a gene responsible for wings, instead of a haltere, what will happen is that Hox protein will turn around and as a transcription factor activate the wing gene. So instead of having one wing, this particular fruit fly will have two wings. Now, it turns out that the homeotic genes have been conserved and are important in evolution. The question is, how do you think that all happened? Well, let's suppose that way back when, we're talking about the ancestor to all animals, and it contained an ancestral Hox-like gene on the chromosome. Now, somehow, some way, that particular gene was duplicated. Okay, so instead of having one, there are now two. And now there are mutations in perhaps the second Hox-like gene, and it takes on a different function. So now we have two Hox genes. They produced homeotic proteins, but they have different functions and control other genes that control other structures. So there may indeed be other duplication events. And so it's thought that there was an ancestor that actually had 4 Hox genes. And now we have a divergence in which we have jellyfish over here. It is our best guess is that the jellyfish lost 2 of those Hox genes because there are only 2 Hox genes or homeotic genes found in jellyfish. However, over here, when we look at an ancestor to bilateral animals, it is estimated that there are 7 Hox genes, so more duplication has occurred. And you can see now what has happened to those 7 genes over the course of evolution. In the case of roundworms, some of the Hox genes have been lost because they only have 5. Arthropods have actually duplicated 1, and so they have 8. Mollusks actually have 11 Hox genes. Echinoderms actually have 12. And then when it comes to cephalochordates, what happened in the course of tetrapods or vertebrates is that this group somehow the entire group was actually duplicated and now can be found on not one, but 4 different chromosomes. So if we look at this particular chromosome, it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 Hox genes. The one below it has 10. The one below that has 9. And the one below that also has 9. Okay? And what they've also found out is that if this particular Hox gene right here controls a structure on the head, then these two also, let's go back. These two also, these two also control structures in the head. And that if this one controls, if this one controls like, digits on the foot, then all 4 of these will somehow have an impact on digits on the foot. Okay, and remember, as we go from left to right, you're looking at Hox genes that control things in humans from your top to your head down to your toes, so to speak. So nearly every animal that has been tested does indeed have homeosequences in its DNA. And the more closely related two animals are, the more that their Hox genes are going to be alike. It also tells you that this gene is extremely important and has been conserved over time. They think that the first duplication happened about 1 billion years ago, and that the ancestor to all animals had at least 4 Hox genes. And I've already mentioned that bilateral animals have 7 Hox genes. And that if you're looking at the course of evolution, the one thing we can say is that the large diversity, animal diversity, is based on 2 simple ideas. That bodies are made out of repeating units or segments, and that there are genetic programs for building structures on those segments. So looking at something very simple like a millipede, each one of those segments has 2 pairs of legs. Something happened to the Hox gene so that down here, instead of having 2 pairs, there's only 1 pair. So that's the difference between millipedes and centipedes. Looking over here at brine shrimp, we can see that we have modified legs that are used for swimming. And then we can see over here on the abdomen, we don't have any. But then when we look at the shrimp, the gene has been modified so that not only do you have those genes like the brine shrimp, but you also have some swimmerets in the back. We can see that over here in a lobster, that particular appendage has been modified to form a pincher, if you will, which you can also see in the scorpion. But now, by the time we get to insects, we don't have any legs on the abdomen area at all. Instead, all the legs are located up here on the thorax. So what we can say is that this is an example of a gene that has been duplicated, modified, and has a lot of impact on different kinds of structures, which is what evolution does. Evolution very seldom creates from scratch. Instead, what it does is that it really edits. So now let's look at what happened with homeobox genes and vertebrates. So if this represents those genes on a fruit fly, and we're going from the head back towards the posterior, we also find that same sort of structure from in animals. However, if you're looking at vertebrates, what we see is that this entire group has been duplicated and now can be found located on 4 different chromosomes. All of the genes, all of the homeobox genes are similar on those 4 groupings, but each is slightly different. Similar genes in different clusters, by the way, are called paralogs. We also can see now here is an example of what Hox genes are controlling or influencing certain vertebrae in a mouse. And while it's obvious that mice and other vertebrates are not as segmented as something like an arthropod, certain regions of their bodies actually are. Invertebrates, with all their associated muscles and bones, actually grow from repeating units in the embryo called somites. The Hox genes, usually in combinations, help define the somite identity, directing them to develop differently depending on where they are in the body. Just like the Hox genes in arthropods direct segments where to grow legs, wings, or antenna, the Hox genes in the vertebrates directs the segments to grow ribs or not to grow ribs, or bones that fuse together to form the sacrum. Experiments in mice show how the Hox genes affect vertebra identity. In the mouse embryo, the Hox gene 10 turns on the rib program. These genes are normally active in the lower back where the vertebrae don't grow ribs, and inactive in the mid-back where the vertebrae do grow ribs. When the Hox10 paralogs are experimentally inactive, the vertebrae of the lower back turn around and they grow ribs. Something similar may have happened in nature. In snakes, the Hox10 genes have lost their rib-blocking ability, which may explain why they grow ribs from all the way down from their head to their tail. So the Hox genes, they have found out, play many more roles in vertebrate development. They help to specify the difference between an arm and a leg, as well as a pinky and a thumb. In the nervous system, their expression and segmented embryonic structures are called rhombomeres, and they direct the development of different regions of the brains. Hox genes are a fascinating example of how a single gene that did something well was copied and repurposed through mutations. All right, so let's talk about other things that can affect development. So here is a diagram of how muscles develop. First off, we have an embryonic precursor cell, and within that cursor cell, there is a master regulatory MyoD gene called MyoD, and right now that is turned off. And the other genes that are responsible for making muscles are also turned off. Well, there are some neighboring cells that will send a signal to this embryonic precursor cell. That signal may be a transcription factor, but at any rate, it causes a cascade effect, if you will, and it causes this master regulatory gene MyoD to be activated. So now it's going to make a messenger RNA. That messenger RNA is going to make the MyoD protein, which turns out to be a transcription factor. Now, that transcription factor is going to turn around and activate another gene, which will turn around and make another transcription factor, and that transcription factor will actually turn around and activate 2 muscle-building genes. And so when these genes are activated, it starts producing myosin, which is a muscle protein, and other muscle proteins. And in addition to that, the transcription factor will turn around and go to a cell cycle gene and actually block the production of cell cycle proteins. And basically what it's going to do is it's going to stop mitosis. So now what starts to happen is you're making these muscle proteins, and mitosis is taking place, but no cytokinesis. So what you have is you're going to make a long cell with lots of various nuclei in it, a multinucleated cell, and inside of there, you're going to organize these fibers, muscle fibers, and now this particular cell is becoming differentiated, okay? It is becoming a muscle cell. So up here, when this embryonic precursor cell received the signal from a neighboring cell, and this particular regulatory gene was turned on, at that point in time, that particular cell is what we call determined. Its fate is determined, and it's going to differentiate into an adult muscle cell. At this point in time, it's called a myoblast, but it will indeed differentiate into a muscle fiber, a fully differentiated cell. So let's look at another aspect of regulation and development, and that is the fact that development often involves the death of cells. And the type of death that it usually involves is called apoptosis. So here's a picture of a mouse's paw, and at first, this just looks like a plate of tissue, and you can kind of make out maybe some some bones developing, but this webbing, this tissue in between the digits is going to degenerate. These cells are going to die, and they're going to die through the process of apoptosis. Eventually, all that webbing is going to disappear, and we're going to now have digits, and we're going to have a paw on the mouse. They have been able to identify the genes responsible for apoptosis. And these genes are similar in both vertebrates and invertebrates, such as our roundworm or a nematode. And what is also interesting is fungi, such as yeast, also have these genes. That should indicate to you that this particular gene, apoptosis, is a gene that has been conserved and is an ancient gene. So let's look at how it occurs. The cell below is a normal leukocyte, and the cell above it is a cell that's undergoing apoptosis. It's a programmed cell death. We find it in infected cells, cells undergoing development, like we just showed you with the paw of the mouse, and cells that are aged and no longer functional. So what happens in apoptosis is that there are cellular agents that go about the process of digesting or breaking down the DNA. And then there are other enzymes responsible for breaking down the organelles. The cell then starts packaging up bits and pieces of the cell. And these little vesicles, these little membrane-bound vesicles are going to be sloughed off, so that now the leukocyte, if it's around, can undergo phagocytosis and take that in there and digest it and get rid of it. But what that also means is that this cell is going to die. So here is an example in ducks versus chickens. And the concept is called heterotrophy. Heterotrophy is the changes in spatial expression pattern of a developmental gene. So the feet of all bird embryos start off being webbed. It turns out that there's a gene called the morphogenic protein 4, bone morphogenic protein 4, which is BMP4, and it's responsible for cells undergoing the process of apoptosis and dying. Now, both ducks and chickens have that gene. But it turns out that there's a second gene called gremlin. And what gremlin does is it produces an inhibitor protein that interacts with the protein expressed by the BMP4 gene. And basically what it does is it inactivates that protein. So if you're looking at ducks, the gremlin protein is going to inactivate that protein. What will happen is the webbing will stay intact and ducks will have webbed feet for swimming. But if you're talking about a chicken, the BMP4 protein remains active and it stimulates apoptosis in that webbing area so that chickens do not have that webbing. All right, so now let's talk about something else that can affect development, and it's called heterochrony. Heterochrony is the regulation of developmental stages by changing the duration of the developmental process. You and I both know that a big difference between giraffes and humans is that a giraffe has a much longer neck. You probably would hypothesize that if you counted the number of vertebrae in the neck of the human and the neck of the giraffe, you probably would hypothesize that there has to be a lot more vertebrae in the neck of the giraffe than in the neck of the human. But lo and behold, both of them have 7 vertebrae in the cervical region of their backbone. However, there's been a delay in the timing sequence with the giraffe. It turns out that there are some cells called chondrocytes, and chondrocytes are cartilage-producing cells that lay down the structure of a bone. So once the chondrocytes have completed the structure of the bone, there's a signal that results in the death of the chondrocytes, followed by the calcification of the bone by accumulation of calcium salts. In the giraffe, the death of these chondrocytes is delayed, and because it's delayed, what happens is that now these vertebrae are elongated compared to a human. Here's another example of heterochrony, and it happens in zebras. One of the differences between the plain zebra and the grizzly zebra is that the grizzly zebra has more narrow stripes. And it's thought that the genes that are responsible for the stripes are delayed in the plain zebra, resulting in wider stripes. As we have mentioned, timing is extremely important in development. So now we want to talk about how neighboring tissues can also affect morphogenesis. So we have an interaction of 2 layers of cells. One layer of cell is on the outside, and it's called the epidermis. It's the skin, if you will, but it comes from germ tissue called ectoderm. On the inside, it also turns out that ectoderm gives rise to the nervous system. And so as the brain is developing, there's this little outpocketing of some tissue from derived from the brain called the optic vesicle. As it gets close to this ectoderm, that ectoderm causes that outpocketing to fold inward, okay, to give rise to the optic cup, if you will. Now, the optic cup will turn around and interact with this epidermis and cause it to fold inward. And eventually, it's going to induce that epidermis to form a structure we know as the lens inside the optic cup of the eye. So even when the optic vesicle was removed, okay, so if you take this right here, and if you are able to remove it and put it underneath the epidermis of the back, what will happen is that the epidermis of the back and the interaction of the optic vesicle will turn around and cause the back epidermis to form a lens. So the interaction of neighboring tissues can indeed have a strong influence on development. All right, so let's talk about another example. It has to do with gender determination. We know that the reason there are males and females is because males have a Y chromosome. And that Y chromosome is responsible for the development of the male testes. Now, if something, by the way, goes wrong with the Y chromosome, by default, you're always going to have a female. So let's look at what happens. First off, you're going to have a primitive gonad. And that primitive gonad right now has the ability to turn into either a testes or has the ability to turn into an ovary. And if you are a female, then that primitive gonad will turn into an ovary. And if you're a male, that primitive gonad will turn into a testes. The question is, how does that work? Well, it turns out that on the Y chromosome, there is a gene called the sex-determining region Y gene, or they simply call it the SRY gene. And that is responsible for the development of testes. And if for some reason it's not working, then you're going to have, by default, the development of an ovary. So let's look at what is going on. So here is this primitive gonad that we spoke of. And as we look at the embryo, we also find out that there are two different primitive ducts, if you will. In blue, right over here, we have something called the Wolffian duct. And the Wolffian duct is going to develop into, under the right conditions, the epididymis, the vas deferens, the seminal vesicle, and the ejaculatory duct. If it is destined to be a female, then this particular duct over here is called the Mullerian duct, and that's going to develop into fallopian tubes, the uterus, and the upper part of the vagina. So how is it that the embryo knows whether it's going to develop the male gonads or the female reproductive system? Well, this is the way it's going to work. If it is a male, the SRY gene is going to have produce a protein or a factor, if you will, that has an effect on that primitive gonad. The primitive gonad is going to develop into a testes, and inside that testes, there are two different types of cells. There is the Leydig cells, and the Leydig cells themselves is going to produce a hormone, testosterone. Now, testosterone is going to interact with the Wolffian duct and produce all those male parts for the male reproductive system. Now, inside the primitive testes, there's also the Sertoli cells. Now, the Sertoli cells is going to produce a Mullerian inhibiting substance, MIS, and that has the effect of going to the Mullerian ducts and causing them to degenerate. So under the influence of the SRY protein, which is made by the SRY gene, you're going to have the development of the male reproductive system. Now, let's suppose we're talking about a female. Well, if we are talking about a female, first off, there's no SRY gene, so there will be no SRY protein. So the gonad will go ahead and develop into an ovary. And if it develops into an ovary, there are no Leydig cells, so therefore no testosterone. And what will happen is the Wolffian duct will simply degenerate. Now, because it's an ovary, we also have no Sertoli cells. So that means there is no Mullerian inhibiting substance. And so therefore, because there's no inhibiting substance, the Mullerian ducts will develop into the female reproductive system, producing the uterus and the fallopian tubes and upper part of the vagina. So hormones can indeed have an effect on development. Now, as we've mentioned, as cells become differentiated and undergo the process of pattern formation and morphogenesis, it appears that if a cell no longer is pluripotent or totipotent. Once it becomes determined, this fate is sealed and you can't turn it into another type of cell, nor can you create another organism from it. Like you can't take a skin cell, one of your skin cells, and clone you and make another you. And that's what we have always thought. Da-da-da-dum! I need to tell you that science fiction is now here. They have indeed taken an adult cell, differentiated cell, and been able to clone a sheep. And that happened in 1997 at the Roslin Institute in Scotland. And this is the way they were able to do it. First off, this is the animal we're going to clone, and you'll notice that it has a white face. And the other two sheep that we're going to be using has a black face. So to make sure that we cloned that particular sheep, we want to look at the offspring and make sure it has a white face. So just a heads up. So what they did is they took some cells from this sheep from the mammary gland. So the cells are determined, they're differentiated, they're adult cells, and in theory, you should not be able to take those cells and make a clone. Well, what they did is they grew them up in a culture dish, and they basically kind of starved them. And by starving these cells, they were able to make them revert to an embryonic or a totipotent state, if you will. Now, the next thing they did is over here, they need an egg. And so they took it from a sheep that I guess you could say the egg cell donor sheep. And they removed the nucleus from the egg. And what they did now is that they fused this cell, which is diploid, to the egg cell. And then they basically gave it a little bit of a shock to make it think it had been penetrated like fertilization. And so it kind of jump-started the cell thinking, oh my gosh, I'm an egg and I've been fertilized and it's time for me now to start to develop. And that's exactly what happened is it started to do mitosis. And so it got a little tiny early embryo. What they did now with the embryo is they took a number of them, but they implanted them into another sheep that was a surrogate mother. And you'll notice it also has a black face. So they implanted the embryo into the uterus of a third sheep. And lo and behold, that surrogate mother did indeed become pregnant and then turned around and gave birth to a little lamb that they named Dolly. And when they looked at the DNA of Dolly, it turned out to be identical to its mother. And so they were able to clone the sheep. Now you may think that is really cool, but it does turn out that over the course of time, there were things that happened and the DNA, if you want to call it becoming old, or as they said, became imprinted or what have you. But Dolly ended up having to be euthanized at a little earlier age than normal because it came down with a lung disease, and they also noticed that it had arthritis of an older person. And so this was not exactly 100% healthy sheep. Since that time, they have been able to clone a number of different animals, including a cat at Texas A&M, the little kitten they called Copycat, CC. They have been able to clone other kinds of vertebrate. At this point in time, we are not aware of any human that has been cloned. Because of the ethical issues that go along with it. So the point being, once upon a time, we used to think that cells, once they became determined, once they became differentiated and specialized, we no longer, even though they had all the DNA necessary, we no longer could clone from that particular cell. And now what we have been able to do using the process we just described is we have indeed been able to clone a cell, an adult differentiated cell. All right, so I hope that this has shed some light on the process of development and that you understand how important genes are and transcription factors and how important cytoplasmic determinants are in development. So I hope that you do well on your AP exam. You make a 5, and we will talk to you later. Take care. Bye.